Alright, in this chapter we start to look at cells, so we're actually looking at something that is alive, finally. So the cell is the smallest living unit, and just to give you an idea of the size, here's a scale. Your most plant and animal cells are right in this region here. Your bacteria are going to be smaller in general. Your mitochondria are also going to be a little bit smaller than the bacteria. This range here are things that are visible by the light microscope. And then the human egg is right here. This range here is what is going to be considered visible to the unaided eye, giving the relative size in millimeters, centimeters, and meters here. The human egg is just borderline on this size. It's the largest human cell that can almost be seen with the naked eye. And then down below here, you've got things that need to be visualized with an electron microscope. And then there are things like some of your proteins are large enough, but lipids, your small molecules and atoms are too small for the light microscope as well. So since the cells are too small to see with the naked eye, we use a microscope. What we use and what most classrooms use are the light microscope. It's going to pass visible light through the specimen and then through a glass lens, and then it refracts, and it can magnify the specimen up to about 1,000 times. Some cells we don't need to view at 1,000 times magnification. The magnification is going to be the ratio of the object size to its real size. So how much bigger does it appear when you look at it underneath the microscope? The resolution is the measure of clarity of the image. That can be defined as the minimum distance that you can distinguish two separate points as two separate points. With the light microscope, it's at about 0.2 microns or micrometers, or 200 nanometers. What limits this is the shortest wavelength of the light used, not necessarily the microscope, but it's just the properties of visible light itself. Contrast is going to be added in with stain. This is going to accentuate the difference in the parts of a sample. It makes it a lot easier to see when you can stain the cells. Cell walls were first seen by Robert Hooke in 1665. Anton van Leeuwenhoek was a Dutch lens maker. He made the first light microscope. His microscope actually was really not much more complicated than a magnifying glass. Most organelles, your membrane-bound compartments in the cell, are going to be too small to actually see with the light microscope. What you're going to see under a light microscope is going to be the cell. You can see the cell membrane. You can see the nucleus, a lot of times the nucleolus, the genetic material, whether it be chromosomes or in the form of chromatin, and that's about it. So the electron microscope was introduced in the 1950s. These focus beams of electrons through the specimen or onto the surface. Their theoretical resolution is about 0.0002 nanometers, but in practice it's still only about 2 nanometers. It's going to reveal a lot more in the cell, but the preparation kills the cells. You actually have to use toxic stains for electron microscopy, and they also have to be prepared in a vacuum. And then on top of it, electron microscopes are not something that's readily available to most people. They are quite expensive. So your ultrastructure is the cellular anatomy, and this can be seen with the electron microscope. These images here give you some ideas of the differences between what you would see with different types of microscopes. Right here is a bright field microscope. It's a light microscope, unstained. You can see the cells. This is with phase contrast, and then this is with a stained specimen right here in the middle. You can see that the stain makes a tremendous difference. Most of the time you will stain something because it is so much easier to see it when it is stained. So with the electron microscope, you've got the SEM or scanning electron microscope. It's going to show the image of the object's surface. And then the transmission or TEM electron microscope will show the internal structures. So over here on the left, this picture is showing a scanning electron microscope picture, and the one on the right is showing a transmission electron microscope picture. Cytology is just the study of cells. So if you want to study parts of cells, you can actually do cell fractionation. And with this, you take the cell apart and you separate out the major organelles and the subcellular structures with the centrifuge. This way you can just study the components you want in bulk. So they do use a blender a lot of times in a lab. It's just like a regular kitchen blender to grind up the cells, and then you use a centrifuge. This is not really a great picture of a centrifuge. Generally, they are contained in either like almost a bucket or a bowl or a box of some sort, because they do spin around very fast and you would not want to touch something moving that quick. You could easily lose a finger doing that. They do need to be balanced where you have things opposite each other. Otherwise, it's spinning at that rate. They will tend to walk off the counter. It's a lot like a washing machine, that you put a pair of shoes in there and you have a big towel, laundry wads up on one side, and next thing you know, your washing machine's starting to walk across the room. So the two major categories of cells are the prokaryotes and the eukaryotes. What is going to be the difference is the internal membrane-bound compartments. Both cells are going to have plasma membranes. It's a selective barrier around the outside of the cell. Here is an image of the plasma membrane. All of these little small gray and yellow pieces represent the phospholipid bilayer. It's a bilayer. There is two layers of it, and you've got the fatty acid tails pointing towards the middle from both sides. Then you've got proteins that are embedded in the surface, and then you can have carbohydrate side chains and various other things that are attached on the outside. So what this creates is a region on the outside of the cell that is hydrophilic, a region in the middle of the plasma membrane that is hydrophobic, and then the inside of the cell can be hydrophilic as well. Your cytosol is a semi-fluid-like substance that's inside the cell. It's going to contain the cell's components. The cytoplasm is going to be all of the contents except the nucleus, which sounds very similar. The way I remember the difference is cytosol is like pine sol, which is a liquid. Not the greatest association to make, but it does work for me. So common organelles that you see in all cells are going to have your chromosomes that will carry the genes, and all cells are going to have ribosomes that they use for protein synthesis. The eukaryotic DNA is mostly in the nucleus. There is some extranuclear DNA, but the vast majority is in the nucleus of the cell, and it's going to be surrounded by a double membrane. So it's like this phospholipid bilayer, but there are two phospholipid bilayers. Your prokaryotic DNA is in the nucleoid region. There isn't a separate membrane-bound nucleus, but it's not freely floating around either. It's concentrated in one particular area of the cell. So this is showing a prokaryotic bacterial cell here with its nucleoid region all being condensed in one area. So the cytoplasm is the interior of the prokaryotic cell, and it's the area between the plasma membrane and the nucleus of a eukaryotic cell. So it does not include inside the nucleus. In general, the size of eukaryotic cells is much larger than that of prokaryotic cells. One important thing to note with cells is their metabolic requirements are going to limit their size. Just because you have a bigger organism doesn't mean you're going to have bigger cells. Instead, you will have more cells. And this is because of the surface area to volume ratio. Smaller cells have a greater ratio. Because the surface area is important for what can get in and out of the cell, if you have too much volume for the amount of surface area you have, you're not going to be able to get nutrients in and waste out. So here with this small little square, it's showing you've got your surface area and volume 6:1 for a ratio of 6. When you make this bigger cell here, it goes down to 1.2. You can have the same volume here made up by multiple small cells and still preserve that surface to volume ratio. So the internal membranes are going to separate things into organelles. They will also facilitate and participate in the metabolic functions in the cell. So when we look at where your genetic material is in the eukaryotes, it's in the nucleus. This is where most of the genes are. It's surrounded by a nuclear envelope. It's a double membrane of lipid bilayer with its associated proteins, and it's also going to be perforated for pores, so there are some things that are going to need to go in and out of a nucleus. The pore complex is going to have the inner and outer membrane be continuous with the lip of the pore. And then you'll have intricate pore complex that will line the pores. And this is going to help regulate what goes in and out of the cell. Most are going to be proteins and RNAs. The nuclear lamina, this is a net-like protein filament that's going to be on the nuclear side of the nuclear envelope. It's going to help in maintaining the shape. The nuclear matrix, this is going to be a framework of fibers that are going to penetrate throughout the nuclear interior. By having this lamina, it's going to help hold the shape, and the matrix just helps to provide a little bit of support, holding the shape and anchor things in place a little more inside the middle. Your chromosomes are the organized units of DNA. These are going to be what you form during cell division. There is a characteristic number of these in each eukaryotic species. Just as a side note, more chromosomes does not necessarily mean a more advanced species. Humans have a diploid number of 46, but there are ferns and unicellular species that have much greater numbers of chromosomes than we do. So chromatin, this is the complex of the proteins and the DNA in a loose diffuse mass. This is how it will be most of the time when it's not formed into a chromosome. This way the enzymes are able to get in and access the DNA. Your nucleolus, or nucleoli if it's plural, is going to be seen in cells that are not dividing. These are going to synthesize your rRNA or ribosomal RNA. They'll help import proteins in to assemble your large and small ribosomal subunits, and those subunits are going to exit through the nuclear pores into the cytoplasm to form your ribosomes. It also helps to regulate your cell division. So the mRNA, or your messenger RNA, this is synthesized in the nucleus. So remember, this is our working copy of the DNA that we're going to send out to the cytoplasm for the ribosomes to translate, so you can protect that one and only good master copy. So here you can see where you've got, this is the nucleus of a cell, you've got the chromatin and a nucleolus. So the ribosomes are your protein factories. They're a complex of ribosomal RNA and protein, so they're actually made partially of RNA, and we call it rRNA, or ribosomal RNA, because it's in a ribosome. Their job is to carry out protein synthesis, so they're going to be more abundant in cells that do a high amount of protein synthesis. Some of them are going to be free-floating, others are bound. The free ribosomes are going to be suspended in the cytosol. They're usually involved with functions in the cytosol. A lot of times they catalyze the first steps of sugar breakdown. Your bound ribosomes are going to be attached to the endoplasmic reticulum, or ER, or nuclear envelope. The proteins are inserted into the membranes. There can be packaging within the organelles, and then they will help to export secretions. This is just showing here your large and small subunit of a ribosome. Over here on the left, you can see some of them are free ribosomes, and then some of them are bound to the endoplasmic reticulum. The endomembrane system is going to be a series of different membranes to carry out various tasks such as protein synthesis, transport, involved with metabolism, moving lipids, and detoxification of poisons. It will include various membranes that are not necessarily identical in structure or function. One of the things they can do is create vesicles, which are just membrane-bound sacs. By putting substances in a sac, it's a way of isolating it from other parts of the cell that you may not necessarily want it to interact with. So the endoplasmic reticulum, or ER, is an extensive membrane network inside the cell. It makes up over half of the total membrane in eukaryotes, and it consists of a series of tubes and sacs. So the cisternal space is going to be the internal space, or the lumen, and it's separated from the cytosol by the endoplasmic reticulum. And it's also continuous with the nuclear envelope. So if we go back up to this picture here, you can see where you've got endoplasmic reticulum, and here is the point where it is continuous with your nuclear envelope. Here you can also see that you've got the pore complex where it's going to be continuous as well with that phospholipid bilayer that's a double membrane. So your smooth ER is going to lack the surface ribosomes. It's important in making enzymes that are used for lipid synthesis, steroids, and phospholipids. It's involved with detoxification of drugs in the liver. A lot of times what it will do is attach a hydroxyl group to increase the solubility and facilitate flushing it out. One of the ways that we develop a tolerance to a lot of drugs is as you take them, you'll start to produce more smooth ER that will begin to break it down quicker. So it takes a higher amount of the drug to achieve the desired effect. So it can store calcium ions. It's called sarcoplasmic reticulum in muscle cells, and that's what it does, is store the calcium ions that you need for muscles to be able to contract. The rough ER is going to have ribosomes on the surface. These are going to make more of your protein secretions like insulin and glycoproteins. It will make these secretory proteins and keep them separate from the cytosol. They can be wrapped up in transport vesicles. It's also involved with membrane synthesis. It can add proteins and phospholipids to its own membrane as well. So here again it's showing it being continuous with the nuclear envelope. Here's a transport vesicle being released off the side. So the Golgi apparatus is a series of flattened membrane sacs or cisternae in stacks. Its job is to modify and store proteins and then send them off to other destinations. It's extensive in secretory cells, and you've got a definite polarity. There's an in and an out, or an entrance and an exit with this. It's got the cis and trans phase. The cis phase is your entrance. It's going to receive the vesicles. The trans phase is going to give rise to vesicles that have been modified in transit. I like to think of it like a UPS store. You go in the front door, you bring in your item you want to have packaged up. You leave it with them while it's in the store, it gets packaged up, gets a label put on it, and it's going to go out the back door into the UPS truck. So it's been modified in transit as you've moved it through the store. One of their jobs is to modify carbohydrates and the glycoproteins. They can also alter membrane phospholipids and manufacture certain macromolecules and secrete polysaccharides. So the fact that things come in on the cis phase and exit out the trans phase, we call it the cisternal maturation model. And that it's going to modify the cargo as it goes. It marks its products with molecular ID tags so the cell knows where to send things. Here we're just seeing something come in through the cis phase and exit through the trans phase of a Golgi apparatus. So the lysosomes are membrane-bound sacs of hydrolytic enzymes. Some means body or cell, and lyse is to split something open. So these enzymes are going to help digest macromolecules in animal cells. Inside, you're going to have proteins that are going to line and protect the lysosomal membrane so it does not digest itself. So one thing it's used for is phagocytosis. When you have a food vacuole or a smaller organism that has been engulfed, it will form and fuse with a lysosome, and the enzymes will digest it or break it down into the monomers so it can be used as nutrients for the cell. With autophagy, here the cell uses hydrolytic enzymes to recycle its own material. Sometimes it does have materials that it just wants to recycle and break down. So Tay-Sachs disease, this is a lipid digesting enzyme that is missing in these individuals or inactive. What happens is this will allow lipids to accumulate in the brain, which will impair its function. It is rare, which is fortunate because it does not have a good outcome. It's generally fatal. So vacuoles, one type is a food vacuole. These are formed by doing phagocytosis of food. Another type is the contractile vacuole. These are going to pump excess water out of the cell. These are going to be found in some of the freshwater protists. They're constantly taking water in, so they have to have a system to constantly pump water out. It's like having a boat that has a leak in it. Unless you're constantly pumping the water out, it's going to fill with water and sink. The central vacuole, this is found in plants, and a lot of times it's formed by the coalescence of smaller vacuoles. It can carry out hydrolysis, it can hold reserves of repository ions, be involved for disposal of metabolic byproducts, can contain pigments or poisonous compounds. So in the morning, most of you are probably consuming something that is stored in a central vacuole in a plant. One example would be caffeine. So the mitochondria and the chloroplasts are involved with the energy systems of the cell. The mitochondria is where we're going to do cellular respiration, and the chloroplast is where we do photosynthesis. We'll go into both of these processes in more detail a little bit later. These are enclosed by membranes, but they're not actually part of the endomembrane system. These membranes were made by free ribosomes. These organelles also contain a small amount of DNA and are semi-autonomous. They can grow and reproduce within the cell. So how these came about is what we call the endosymbiont theory. So you had a cell that ended up engulfing, in the case of a mitochondria, an oxygen-using non-photosynthetic prokaryote. That prokaryote lost the ability to survive on its own, and eventually it became dependent on the other cell as an organelle and became the mitochondria. You also had a cell that would engulf a photosynthetic prokaryote, which became the chloroplast. So there is a lot of evidence for this. For one, both the chloroplast and the mitochondria have their own genetic material. You're going to see ribosomes that are more similar in style to the prokaryotic cells than the eukaryotic cells that they're actually in. And they also have a double phospholipid bilayer as the membrane. So you had the original membrane that was around them, an additional membrane that was formed and not lost when they were engulfed. Peroxisomes, these are oxidative organelles. They're not part of the endomembrane system either. So the mitochondria, these are present in nearly all eukaryotic cells. It's enclosed by two membranes of phospholipid bilayer. The outer membrane is smooth and the inner folds to form the cristae. So in the inner membrane space, it's the region between the inner and outer membranes. And then with the mitochondrial matrix, this is going to be enclosed by the inner matrix. This will contain a lot of enzymes and DNA, and it's highly folded for a lot of surface area. And we actually need that surface area of those membranes in there to be able to pass electrons down in the process of creating energy. So the chloroplasts, these are used for photosynthesis. They're a specialized member of the plastids. The amyloplasts are one variation that will store starch. Chloroplast stores pigments. Inside we've got these thylakoids, which are interconnected membrane sacs. They will have stacks of these thylakoids that are going to form granum or grana. The stroma is the fluid outside the thylakoids. It's going to contain the DNA, ribosomes, and enzymes. So the peroxisome, this is going to be a specialized metabolic component that is bound by a membrane. It produces hydrogen peroxide. So some of it is used to break down fatty acids, also to detoxify alcohol in the liver. The glyoxysomes, these are specialized fat-storing organelles in the tissues of plant seeds. What they will do is they will convert the fat in the seed to sugar to help feed the seedling. So nuts and seeds tend to have a lot of fat content in them. And so when they fall down to the ground, when you're not eating them, what you'll have is the new little plant start, and then you can use the fat from that seed and turn it into sugar to give that plant a little bit of food before it's able to produce it on its own. So the cytoskeleton is a fiber network in the cytoplasm, and it's going to organize the structure and activities in the cell. So the peroxisome, let's see where we left off. The cytoskeleton, this is your fiber network in the cytoplasm, and it's going to organize and provide structure and activities inside the cell. So it helps to provide mechanical support for shape. It will provide mobility by interacting with the motor proteins. This is what happens with your structures like the cilia and the flagella that you're going to use for motility. The cilia are more like hair-like structures around the cell. Flagella is more of the whip-like tail. It's going to provide transport pathways inside the cells. It will move the plasma to do phagocytosis and regulate biochemical activities in response to mechanical stimulation. So it can actually sense when something is touching the cell and cause a biochemical response. This image here is just showing a vesicle with a motor protein moving along the microtubule of the cytoskeleton, a lot like a train moving on tracks. So your components, you've got, the first is the microtubule. It's the thickest fiber. It's made up of the protein tubulin that provides shape and support. It's also going to provide tracks for the organelles to move on. It will help to guide the secretory vesicles. The centrosome, this is present in animal cells. These organelles are not well understood. It's going to be in the region where the microtubules are going to grow out of. It's involved with cell division. We know it's not mandatory for cell division because plant cells do not have them. So the centriole is going to be within the centrosome, and it's composed of 9 sets of triplet microtubules that are arranged in a ring. So before animal cells divide, these will replicate, but we don't see them in plants or yeast. So here you can see that you have the 9 sets of triplet rings, and this is the cross section. So cilia and flagella, these are going to be microtubule containing extensions. These are going to project from some cells to propel them. The flagella have an undulating motion that's like swimming. It's going to move in the same direction as the axis. The cilia, on the other hand, are going to work like oars with alternating power and recovery strokes. They extend at right angles to the forward movement, so they're going to be like oars paddling a boat forward. We have different kinds of cilia. The primary cilia will act like a signal receiving antenna and transmit molecular signals. These appear to be crucial in brain function and embryological development. The arrangement, you've got a 9+2 pattern that you will see where it's 9 doublets of microtubules arranged in a ring, and then you have 2 single microtubules in the center. This is present in nearly all eukaryotic flagella and the motile cilia. Your non-motile primary cilia are going to have a 9+0 pattern. It's 9 triplets of microtubules, and they lack the central pair in this type of cell. Your flagella are going to be slightly different in your prokaryotic cells as well. The basal body is going to be what anchors the microtubule assembly. So here you can see the 9+2 pattern, and here's the 9+0 pattern. The dinins are going to be protruding motor proteins that run along the length of the cilia or flagella. These are going to be responsible for the bending movements. So these are going to bend and cause the protein to change shape with energy from ATP. So you can see here when you have them straight and when ATP is present, they will bend and flop back and forth side to side. So the microfilaments, these are solid rods made of the protein actin. Actin is a globular protein that will be shaped in twisted chains. They help give support to the cell shape. They'll give the cortex of the outer cytoplasmic layer a gel consistency. They'll help to bear tension forces when the cell needs to stretch. They're going to be the core of the microvilli that help increase the surface area. For motility, the contractile apparatus of a muscle is going to have actin. It will work with the thicker myosin protein. It's also involved with amoeboid movement. It helps to extend the pseudopodia or create cellular extensions. With cytoplasmic streaming, it's going to cause a circular flow of the cytoplasm within the cells. Just like you have your blood circulate in your body, you're going to have the cytoplasm circulate in the cells. So here, this is an example of your actin filaments with the myosin in skeletal muscle. Here you can see the pseudopodia extending. And then here in this cell, you can see the cytoplasmic streaming where the cytoplasm spins around in the plant cell. Then we have the intermediate filaments. They're larger than the microfilaments. They're specialized to bear tension forces. These will persist after the cell dies. So just like our skeleton persists after we die and other things break down, these fibers will last longer as well. They help make up the nuclear lamina lining the nuclear envelope, giving shape and framework. So we do have some components outside of the cell. We have cell walls that are going to be extracellular in plants. These are going to provide protection, help maintain shape, and prevent excess water uptake. These cell walls are going to be what helps to hold the plant up against gravity and allows plants to get taller. They're made largely of the cellulose. Cell walls are also going to be in prokaryotic cells, the fungi, and in some protists. Cell walls are thicker than plasma membranes. The myofibrils are going to be made up of cellulose that's embedded in a matrix of other polysaccharides and proteins. This will vary in different cell types and species. So you do have strong fibers in the matrix, which make up the ground substance. So the primary cell wall is what you see in young plant cells. It's when it's first secreted, it's relatively thin and flexible. The microtubules in the cortex guide the cellulose synthase and help orient the cellulose deposition to affect the growth pattern. So when you have fresh little plants and you put them in the ground, you have to be really careful with them at this stage like this. If you put too much water on them or the water hits them directly, you're just going to knock them over because the primary cell walls are really flexible. The middle lamella is going to be between the primary cell walls of adjacent cells. This is a thin, sticky layer of pectin that's going to help to glue the cells together. The secondary cell wall gets added between the plasma membrane and primary cell walls in mature cells. Other cells will just harden the primary cell wall. So cells are going to need to be able to communicate with each other, so we'll have the plasmodesmata. They're perforated channels between the cell walls. You'll see these in plant cells. So here down at the bottom, you've got these various plant cells, and these are little plasmodesmata or channels between the cells. So we also have extracellular material in animal cells. It's an extracellular matrix. It's going to have glycoprotein. The most common one is collagen, and it's going to form strong fibers around the outside. So in this picture here, you can see where you've got these collagen fibers. Proteoglycans are small core proteins that a lot of times are linked with carbohydrate chains that form a network where the collagen is embedded. So here you can see the proteoglycan complex in the green. The fibronectins are going to bind the surface cell proteins called integrins into the plasma membrane. So here are your integrins, and here's the fibronectins connecting to it. The integrins are going to span the plasma membrane, and they bind on the cytoplasmic side to proteins attached to the microfilaments. This way you can transmit signals between the extracellular matrix and the cytoskeleton on the inside. This can actually influence genes all the way into the nucleus. So all living cells in multicellular organisms are going to be functioning together, so these intercellular junctions are important. We have the plasmodesmata that are those perforating channels in the cell walls of plants. It allows the cytosol to pass and chemically connect adjacent cells. We have tight junctions. These are going to be tight-pressed animal cell membranes that are bound by specific proteins. It seals around the cells and makes the skin watertight. The desmosomes, these are going to attach the muscle cells to each other, and they're made of the intermediate filaments of keratin. And then the gap junctions. These are cytoplasmic channels between cells that are going to be used to communicate in the heart muscle and skeletal muscle. So overall with cells, we do study them as units, but they are a living unit greater than the sum of the parts because they do interact with each other.